Chapter 15, Regulation of Gene Expression. First, we're going to deal with prokaryotic regulation. Bacterial cells have evolved to only express genes whose products are needed by the cell. That is, that they get rid of inefficiencies and can create proteins when needed and stop creating proteins when it's not needed. And metabolic control happens on two levels. First, cells adjust the activity of the enzymes that are already present. And second, the cells can adjust to the production levels of those certain enzymes. So when looking at prokaryotic regulation, we're going to look at one specific example throughout 5.1. We're going to look at the example of tryptophan. And in this diagram over here, you can see on the left-hand side the regulation of enzyme activity, which would correspond to the first one, and feedback inhibition, which means, if you don't remember, which means that that cycle creates enough of tryptophan that it actually stops itself from creating it once enough of it has been created. And that's one form of regulation. And on the other side, part B, the environment provides all the tryptophan the cell needs, so the cell stops making those enzymes so that it's before those enzymes can even be created to get to part A, or the first, metabolic control. Alright, so now we're going to talk about the operon model. And it was discovered by two scientists in Paris, and it basically states that many genes of a bacteria are switched on and off by changes in the metabolic status of the cell. So we just saw that where multiple genes in tryptophan were affected by one on-off switch. And now we're going to look at two definitions and an actual diagram showing the operon model in action. So an operon is the entire length of DNA required for enzyme production in a pathway, and an operator is a small segment of DNA that acts as the on-off switch, regulating a whole cluster of related genes. So we have the operator here that's controlling all these genes, and this whole thing from here to here is the TRP operon per se. So now we're going to talk about operon repressors. The operon can be switched off by a protein called a repressor, in this case called the TRP repressor, TRP standing for tryptophan. The repressor attaches to the operator and blocks the attachment of RNA polymerase to the promoter, thus preventing the transcription of genes. The TRP repressor is the product of a regulatory gene, TRPR. So if you look at this graphic on the right, you see tryptophan, the co-repressor, bonding with the protein, changing the protein shape, and then the protein shape having a high affinity for the operator and attaching to the operator, and then no RNA is made, thus stopping transcription. So now regulatory genes. Regulatory genes are expressed continuously and are always present in E. coli cells. And the binding of repressors to operons is reversible, and what we're talking about is the tryptophan co-repressor binding to the protein right here. Those regulatory proteins are called allosteric, having two shapes, one inactive and one active. Only when tryptophan binds with the TRP repressor will the protein change to its active state and bind to the operator, because before it had a low affinity for the operator, and once it attaches it has a high affinity and wants to really attach to that operator. So there are two different types of negative gene regulation, repressible and inducible operons. The TRP example that we just looked at was the classic example of the repressible operon, where its transcription is inhibited or repressed when tryptophan binds allosterically to the regulatory protein. Inducible operons, on the other hand, are usually off, but can be stimulated when a molecule interacts with a regulatory protein. And we're about to look at another example, the lactose example, which is the classic example for the inducible gene regulation. So the lac operon example. So lactose metabolism begins with hydrolysis and disaccharide lactose, milk sugar, into its monosaccharides, glucose and galactose. This reaction is catalyzed by the enzyme beta-galactosidase. In the absence of lactose, there are very few of these enzymes, but with the addition of lactose, the number increases thousandfold within 15 minutes. So now we're going to take a closer look at the lac operon example. So here we see the initial state that the operon is in, where the repressor is already active and in the operator where it's in the off position here. And then we're going to scroll down here and see where it's actually producing, where with the introduction of allolactose, the inducer, the protein changes shape and lets go of the operator and creates the enzyme that we were talking about earlier that is created. So this is the on position versus the off position up here, which is the reverse of what we saw earlier. Repressible versus inducible. Repressible enzymes usually function in anabolic pathways, and inducible enzymes usually are found in catabolic pathways. So we looked at these two examples of negative control of genes, which are basically the reverse of each other. And we don't want to get confused that one of them is positive, one of them is negative. They're both negative, and we're going to look into a positive gene control in a minute here. But we have to define that first. So gene regulation is said to be positive only when a regulatory protein interacts directly with a genome to switch transcription on. And we're going to talk about that further on the next slide. Positive gene regulation. Let's look at an example of positive, that's just a gene, sorry about the typo, gene regulation in the lactose operon. So when lactose is present and glucose is in short supply, E. coli uses lactose as an energy source. And how does it know when glucose is in short supply? It determines the glucose supply is low through a reaction with an allosteric regulatory protein, in this case cyclic AMP or CAMP, which accumulates when glucose is scarce. The regulatory protein is called catabolite activator protein, or CAP, and it is an activator. An activator is a protein that binds to DNA and simulates transcription of a gene. So that's what makes it positive gene regulation in this case. So let's look at this diagram explaining the lactose operon example in the positive gene regulation context. So let's focus in right here. So in this context, the lactose is present and glucose is scarce and the CAMP level is high. So the CAMP binds to the inactive CAP, making it active. And this active CAP has a really high affinity for RNA polymerase, so it binds to that and creates the big promoter that we have. And then that promoter starts creating a bunch of mRNA, and that is the positive gene regulation going on in this lactose model that we see right here. Alright, so that was 5.1, and now we're going to talk about eukaryotic gene expression. So a typical human cell might express 20% of its proteins that it can code for at a given time. And the more specialized the cell is, the more specialized the proteins are going to be. And scientists have discovered that the differences between cell types are not due to different genes being present, but different gene expressions. So the differential gene expression is very important, and that causes the cells to be able to differentiate inside a human genome that is the same. So getting different cells from the same genome, that's the really important key of differential gene expression right here. So now we're going to divide this section up into all the different regulated stages of eukaryotes. So this diagram identifies the potential control points where gene expression can be turned on or off, accelerated or slowed down. So This diagram, everything in this purple shaded area is actually happening inside the nucleus, and everything outside of that is happening out in the cytoplasm. So the blue, this one right here, refers to the DNA being affected. Orange, which is right here, the RNA affected, and purple down here will be proteins. So let's start looking at what's going to happen to the DNA in regulation. Regulation of chromatin structure. So remember that DNA is packed into an elaborate complex known as chromatin. This chromatin not only provides structural organization for the DNA, but also helps regulate gene expression in three distinct ways. The first way is that it affects the location of the gene's promoter relative to the nucleosomes where DNA attaches. Two, genes compressed with heterochromatin, a more highly condensed version of chromatin, are usually not expressed. So it's way more difficult for transcription to occur in that section, so it doesn't usually happen. And the third way is that chemical modification to the histone proteins and to the DNA of chromatin may influence both structure and gene expression. So we're going to dive into this third one a bit more because it's a bit more complicated. So histone modification. Histone acetylation is the attachment of groups such as acetyl or methyl or phosphate groups to the histone tails that generally loosen the chromatin, making transcription possible. But these modifications are not permanent per se. And so the way that it's really modifying the histone is it's loosening up the chromatin, making it easier for transcription to occur. Remember that the second way it was regulated was that the heterochromatin was way too compact for transcription to occur. So this is really stretching it out, making it way easier for it to be transcribed and expressed. And DNA methylation occurs mostly in plants and animals and fungi, and it's the attachments of enzymes to the DNA itself. These attachments act similarly to the histone acetylation and make transcription in general less likely. But DNA methylation occurs usually in long stretches of inactive DNA, such as the inactive X chromosome, and so on and so forth. So you see here the methyl groups attaching to cytosine in the DNA structure. So another thing the book went over was epigenetic inheritance, and that is just basically a definition where it's the inheritance of traits transmitted by mechanisms not directly involving the nucleotide sequence. So it's traits that are being passed on that have to do with location and not the sequence that DNA is in. Alright, so the regulation of transcription initiation. Like we saw in bacteria, the regulation of transcription initiation in eukaryotes involves proteins that bind to the DNA and either facilitate or inhibit the binding of RNA polymerase. So in that way, eukaryotes and prokaryotes are similar. So I'm going to do a little bit of review of chapter 14 here and talk about the structure of a typical eukaryotic gene because it's very important for gene expression. So remember that a cluster of proteins called a transcription initiation complex assembles on the promoter sequence at the upstream end of the gene. So one of these proteins, RNA polymerase II, then transcribes the gene, synthesizing a primary RNA transcript. And then RNA processing includes the addition of a poly-A tail, 5-cap, as well as splicing out the introns to yield a mature mRNA. Associated with most eukaryotic genes are multiple control elements, so this is a bit new here. So these control elements are segments of non-coding DNA having particular nucleotide sequences that act as binding sites for proteins called transcription factors that we learned about last chapter. And the transcription factors regulate gene expression. Now I'm going to explore the role of transcription factors in gene expression. As we know, to initiate transcription, eukaryotic RNA requires the assistance of transcription factors. And there are actually two different types, the general transcription factor and the specific transcription factor. So the general ones are necessary for the transcription of all protein coding genes. So an example of that would be the TATA box that we learned about last chapter. And then the specific factor, the specific transcription factors are a little bit weird. It's the interaction of control elements with another set of proteins at specific times. And so I'm going to be going into that and explaining that a bit more in the next slide. So we're going to now talk about enhancers and specific transcription factors. So we have this diagram that may look similar to something you've seen before, but has a little bit extra put over right here. And that's talking about the specific transcription factors I was talking about in the last slide. So in eukaryotes, the rate of gene expression can be strongly increased or decreased depending on the binding of a specific transcription factor to the control elements or enhancers. And the control elements are here and the enhancers are here. So that's really what it's talking about when it's talking about specific transcription factors. So what you might be wondering is, wait, how does something way over here, how does that have any effect on the promoter and then the rest of the downstream sequence? And I'm going to explain that in the next slide. So now I'm going to talk about how activators can influence transcription. So we have three parts here. Two are written down. I'll explain the third one. So activator proteins bind to the distal control elements grouped as an enhancer. So the distal control elements are each one of these three. The activators are binding to it, and the whole thing makes up an enhancer. But that's pretty far away from the promoter and the TATA box. So second, a DNA bending protein brings the bound activators closer to the promoter, where the general transcription factors, mediator proteins, and RNA polymerase II are nearby. So basically, it bends the DNA backwards so that that enhancer and activators are really close to the promoter. And then the third thing that happens is the activators bind to certain mediator proteins and general transcription factors, helping them form an active transcription initiation complex on the promoter, and then the promoter starts to create mRNA. So combinational control of gene activation. So precise control of transcription depends on the binding of activators to DNA control elements. So that's really the way that the specific transcription factors regulate gene expression by telling the promoter how fast or slow to go. And on average, one enhancer is composed of 10 control elements, and the enhancer can only bind with one or two specific control factors. Scientists have discovered that the particular combination of control elements in an enhancer gives it the ability to bind with a gene, not simply the presence of one control element. And that increases the number of combinations significantly. So now I'm going to talk about coordinately controlled genes in eukaryotes. So if you remember all the way back to the beginning of the lecture, coordinately controlled genes in bacteria are clustered into operon that has one on-off switch that controls all of them. Well, in eukaryotes, that can happen and does not happen. The genes are typically scattered across many chromosomes, so that one on-off switch would not work. So the way that they do it is through the presence of very specific combinations of control elements within each gene in the dispersed group. So one analogy you can look at for this is the mailman in your neighborhood. So for outgoing mail, everyone is supposed to put up the little red flag saying, "I have outgoing mail" to the mailman. So he checks that and takes it with him. So that little flag, all of the mailboxes would be all the genes, and every one that has the flag up would be every single gene in that dispersed group, because not all, the way that the prokaryotes do it is they have all the red flags in a line, and you just can't do that with so many genes in prokaryotes. So all the red flags would unify the dispersed group, so the mailman or the chemical signals or whatever is going to express the genes can know to go to every single gene in the dispersed group. So even after transcription occurs, regulation still happens. So we're going to be looking at post-transcriptional regulation. So alternative RNA splicing, which we learned about last chapter, allows many different proteins to be created out of the same primary transcript. And this is also a form of regulation. So we see that from the same genome we get two different proteins, and depending on the environmental factors around, the cell will choose which one to go to, and this is another form of regulation. And yet another form of regulation is mRNA degradation. So each strand of mRNA has a characteristic lifespan, where bacterial mRNA molecules are degraded by enzymes within minutes. In eukaryotes, mRNA typically survive for hours, days, or even weeks. So scientists believe that the relatively short lifespan of mRNA in bacteria enables that bacteria to change its patterns of protein synthesis so quickly in response to environmental changes that it has the upper hand in evolution and doesn't just die off immediately. Another form of post-transcriptional regulation is actually inhibiting the initiation of translation. So translation presents yet another opportunity for regulating gene expression. It's mostly regulated at the initiation stage. So it's blocked by regulatory proteins that bind to specific structures within the 5-cap or the UTR, preventing the attachment of ribosomes, so translation cannot occur, thus inhibiting it and regulating the gene expression. Even after translation occurs, regulation can still happen. So we're going to look into protein processing and degradation right here, which is the last one if you look at the image on the right. So we're at the very end of 15.2 here. So it can involve, regulation can occur at any step involving transportation or modification of a protein. So I've gone through all the different steps of regulation that can occur in eukaryotic cells, and that just ensures that our bodies can react to the environment that we are in quickly and effectively. So I'm going to pass it on to CJ, who will be presenting tomorrow.